Hello everyone. Welcome back. So today we will look at another important topic regarding enzymes, that is the mechanism of enzyme action, what is active site of the enzyme, and what do we mean by Km constant or Michaelis-Menten constant. Okay, so before we begin, if you have not yet subscribed to my channel, please do, as we will be covering the whole of medical biochemistry in lecture series. Okay, so let's begin. What is mechanism of enzyme action? Now, the mechanism of enzyme action has been explained by various theories. Okay, so these are the theories which try to explain the mechanism of enzyme action. Okay, lowering of activation energy, acid-base catalysis, substrate strain theory, covalent catalysis, entropy effect, product substrate orientation theory, Michaelis-Menten theory, Fisher's template theory, and finally Koshland's induced fit theory. So out of these, the important ones are the ones which we will discuss. Okay, so let's begin. First is lowering of activation energy. Okay, so what is activation energy? So activation energy is basically the energy difference between the ground state and the transition state of the reactants. Okay, so we know that reactants are finally converted into products. Okay, and in case of an exothermic reaction, when energy is given out, okay, so this is the energy state of the reactants and this is the energy state of the products. Okay, so in case of exothermic reaction, a reaction is given out. So to achieve the for the reactants to be converted to products, okay, so the reactants first need to achieve a transition state. Okay, so this is the threshold that they need to cross. Let's say it's an intermediate state, which is the transition state, and once the reactants reach this transition state, then they convert into product. Then the process becomes more of like an automatic process. Okay, so the problem is for this reactants to convert from this ground state to this transition state. Okay, so this is known as the activation energy, the energy which is required by the reactants to convert from the ground state to the transition state, and then it leads to formation of product. Now what enzyme does is it lowers this activation energy. Okay, so the enzyme, it lowers the activation energy, so the reactants now require less amount of energy to reach this transition state or the threshold point, after which it can automatically form products. So this is the mechanism of enzyme action. The activation energy is lowered in the presence of enzyme than as compared to without the enzyme. So enzymes, they lower the activation energy. Also, enzymes do not change the free energy between the reactants and the products. Okay, so this is the free energy, the energy which is lost. Okay, so this energy is the energy difference between the reactants and the products. The enzyme, they do not change this free energy or delta G. Okay. Next, coming to Michaelis-Menten theory. Now this was the hypothesis that was proposed by Michaelis and Menten, and it states that the enzyme combines with substrate to form enzyme substrate complex, which it dissociates into enzyme and product. Okay, very simple theory. So enzyme combines with substrate to form as enzyme substrate complex, which is the transition state, and it dissociates into enzyme and product. Okay, so this enzyme substrate complex is the intermediate, and it has weak and non-covalent bond. Okay, we always require a weak bond, because if the bond is strong, then this process needs to be, the time taken to break the enzyme substrate complex will be more. Okay, so we require weak bonds so that this enzyme substrate complex can be easily broken into enzyme and product. Okay, next is one of the most important theories, earlier theory is Fisher's template theory. Now this theory states that the 3D structure of enzyme's active site is complementary to the substrate. Okay, so we know that enzymes are proteins. Okay, proteins are organized as primary, secondary, tertiary, and quaternary structure. So the tertiary structure is the functional structure of protein, and it's a three-dimensional structure. Okay, now the 3D structure of enzyme or the protein is such that the active site of the enzyme, okay, where the action is taking place, is complementary to the substrate. Okay, so if you see over here, this is the enzyme, this is the active site. Now this enzyme 3D structure is so oriented that the active site becomes complementary to this specific substrate. Okay, so similarly you can see over here this particular substrate, the active site is complementary to this substrate, whereas if this is the shape of the substrate or structure of the substrate, then the active site of the enzyme is not complementary. Okay, this is more like a lock and key. Okay, if you have the key is very specific to the lock. Okay, similarly the substrate is very specific to the active site of the enzyme. So that is why this theory is also known as lock and key model. Okay, so this theory however is a rigid structure, does not explain the flexibility of enzyme. Okay, so this theory has the disadvantage that this is this rigid structure of the enzyme, it does not explain the flexibility of the enzyme. Okay, so that is the allosteric effect of the enzyme. So what is allosteric effect, that we will see. Okay, so it also fails to explain the allosteric effect of the enzyme. Okay. Now let's next look at Koshland's induced fit theory. Now here in this theory, this explains the allosteric effect as well as the flexibility of the enzyme. Okay, let us see what that is. The active site of the enzyme undergoes conformational change to accommodate the substrate. Now see over here, this is the active site of the enzyme. Okay, and this is the substrate. Now as the substrate comes near to it, the active site undergoes a excuse me, conformational change to accommodate the substrate. Okay, and ultimately it leads to formation of products. Okay, so this is what is known as Koshland's induced fit theory. Okay, so the substrate binding induces the conformational change to the enzyme. Okay, and why this conformational change? Because the catalytic groups becomes more oriented. Okay, the active site is where all the catalytic groups are. Okay, so when the substrate binds to the substrate binding site, then the active site also undergoes certain changes so that the catalytic groups which are present in the active site, they become more available, they become more oriented. Okay, so the active site is okay, modified by binding of the substrate. Okay. So this explains the allosteric regulation of enzyme. Now what is allosteric regulation or flexibility of the enzyme? It is that the binding of substrate produces certain changes so that at the active site, the enzyme action can be either promoted or inhibited. Okay, so there are certain factors which regulate the binding of substrate. Those are known as allosteric effect or flexibility of enzymes. The enzymes becomes flexible, the active site becomes it changes or accommodates the binding of substrate. So that is allosteric effect of the enzymes. Okay. Next is acid-base catalysis. Okay. Now this involves acid-base interaction like donation or acceptance of a proton or H positive ions at the active site of enzyme. Now if you look over here, this is one nucleotide consisting of a base, sugar and a phosphate group, and this is another nucleotide consisting of base, sugar and the phosphate group which is attached over here. Okay, so this is a phosphodiester bond, okay, between two nucleotides. Now when a ribonuclease is the enzyme which breaks this bond, so when this bond is broken, now what happens, this ribonuclease enzyme contain histidine. Now histidine is the most common amino acid undergoing acid-base catalysis. Now if you excuse me, if you look over here, this is the histidine. Okay, now this histidine ultimately, the H positive ions are taken up by histidine. Okay, and we have the water molecule which is breaking this phosphodiester bond with the help of RNase enzyme. So the H positive ions which is breaking the bond, okay, it breaks the phosphodiester bond, and this H positive ions is incorporated by histidine which is present in the ribonuclease enzyme. Okay, so this is how ribonuclease acts by accepting the H positive ions and causing hydrolysis of the phosphodiester bond with the help of water. So this is how ribonuclease cleaves the phosphodiester bond and how the acid base catalysis is useful or explained. Then there are other theories like substrate strain theory, where binding of the substrate to the enzyme produces strain on substrate and the energy level of substrate is raised. Okay, so when the substrate binds to the enzyme, okay, the substrate becomes strained. Okay, as a result of which the energy level of substrate is raised, which crosses the threshold level or activation energy, and then the enzyme action takes place. Okay, or the reaction takes place. So this is substrate strain theory. In covalent catalysis, the substrate and enzyme bind covalently by positive or negative charge, which is present in ionic groups on the enzyme. So in covalent catalysis, we have there's binding of positive and negative charges between enzyme and the substrate. Then there is entropy effect. The theory explains that enzymes bring together substrate and brings together substrate and decreases the disorder or entropy and increases probability of collision. Okay, so in this case the enzyme is just bringing the substrates together and hoping for the fact that these two substrates will collide with each other and bring about a reaction. So this is known as entropy effect. Lastly, there is product substrate orientation theory. Here again the enzyme keeps the substrate oriented in such a way that the substrates they react and bring about reaction. Okay, so these are the other theories which try to explain the mechanism of enzyme action. Okay, so enzyme basically you can see over here in the first one, it is training the substrate so that the activation energy is achieved. Here the enzymes are orienting the substrate so that the positive and negative charge they react covalently. In case of entropy effect, the enzymes are again bringing the substrates together so that they start reacting together and bring about enzyme action. And lastly, the substrate orientation theory is when the enzymes bring the substrates together in certain orientation so that the reactive groups which are present start reacting. So these were the theories which try and explain the mechanism of enzyme action. Now let us look at active site of the enzyme. Now this is the site where all the action takes place. Okay, it is the region of the enzyme where the substrate binds to the enzyme by non-covalent bonds by realignment of amino acids. Okay, now this is where the site where the catalysis or actually the enzyme action takes place. Okay, it is usually located in a cleft or a crevice. You can see over here, this is the enzyme. Okay, and this crevice or a small part is where the active site of the enzyme is, which is going to bind with the substrate which is having the same structure as that of the active site. Okay. So this is the 3D structure of the enzyme. Okay, and it is so oriented that we have the presence of active site. Okay, and to this active site again we have two types of sites which it contains. One is substrate binding site and the other is a catalytic site. Now the substrate binding site, as the name suggests, that it is going to bind to the substrate. Okay, and why? Because the substrate and the substrate binding site have very specific orientation of amino acids. The amino acids are so aligned in such a way that this particular substrate is going to bind to the substrate binding site which is present inside the active site. Okay, and no other substrate is going to bind. You can see the structure also is complementary. Okay, so it enables the binding of substrate binding site. Now when the substrate binds to this, you have a catalytic site where the reactive groups are present. Okay, where the coenzymes are present. Okay, we discussed about group 1 and group 2 coenzymes. Okay, so the group 1 coenzymes causing transfer of hydrogen ions, hydrogen groups. Group 2 responsible for transfer of groups other than the hydrogen groups. Okay, so which basically consists of the vitamins. Okay, so this is the catalytic site where all these coenzymes are present and where all the reaction which brings about the reaction. So when the substrate binds to this substrate binding site, okay, then this catalytic site is activated. And in case of allosteric enzymes or Koshland induced fat theory, binding of this substrate to the substrate binding site also brings about alignment of the catalytic site, so that the binding is more secure. Okay. So and serine is the most common amino acid seen in the active site. Okay, so this active site, the most common amino acid seen, which is serine. Okay, so that is why we look at enzymes, all serine containing enzymes. A lot of that means the enzymes are having serine in their active site. Okay. Next we look at Michaelis-Menten constant. Okay, so what is this Michaelis-Menten constant? It is basically the substrate concentration at half the maximum velocity of the enzymatic reaction. Okay, so we have an enzyme reaction. Okay, so here on this graph you can see this is the velocity of the reaction and this is the substrate concentration. Okay. Now as we increase the substrate concentration, the reaction also increases. Okay, and up to a certain point, okay, let's say at point C, if you further increase the substrate concentration, the reaction is not going to increase. So we say that this reaction has achieved its maximum potential or maximum reacting capability or velocity of the reaction. Now if we look at the substrate concentration, which is half the maximum velocity, okay, so this is the Vmax we have achieved. Now we take half of Vmax, okay, and we drop a line on the substrate concentration. Then the substrate concentration at half the maximum velocity, that becomes your Km constant or Michaelis-Menten constant. Now why is Michaelis-Menten constant important? For two reasons. One is it is independent of the enzyme concentration. Okay, see over here. Now in this case we have the red line shows enzyme as 1 unit, blue line shows the enzyme as 2 units. So we have increased the concentration of enzyme. Okay. Now when we have increased the concentration of enzyme, now first when the enzyme was 1 unit, the velocity was 100%. Now we have doubled the concentration of enzyme, so obviously the reaction also becomes double the speed. Okay, so 200%. Now in both cases, if we take the Michaelis-Menten constant, that is the half the maximum velocity, here the Km constant remains the same for both enzyme unit 1 or enzyme unit 2. Okay, so Km constant is independent of the enzyme concentration. Okay, it is also the signature of the enzyme. It is constant for the enzyme and the substrate. Okay, so it tells us about the enzyme. It is specific for the enzyme. It is the signature of the enzyme. Okay, so see over here, let's see. It denotes the affinity of the enzyme for the substrate. The lesser the Km value, the more affinity the enzyme has for the substrate. Okay. So let us again see over here. So now if the Km value is more, okay, that means the affinity is less, and if the Km value is less, that means the affinity is more. Okay, so the Km value tells us about two things. One is it is the signature of the enzyme, and secondly it denotes the affinity of the enzyme for the substrate. If the Km value is more, that means the affinity of the enzyme and the substrate is less, and if the Km value is less, that means the affinity of the enzyme and substrate is more. Okay, so this is best explained by the enzyme glucokinase and hexokinase. Okay, both have the same function. Okay, that means attachment of glucose phosphate groups. Okay, glucose to glucose 6 phosphate. Okay, so hexokinase occurs in all tissues. Okay, glucokinase occurs only in the liver. The Km value of hexokinase is 10 to the power minus 2 millimoles per liter, whereas glucokinase is 20 millimoles per liter. Okay, so it is very high. If the Km value is high, that means the affinity is for glucose is low. Okay, now here the Km value is low, that means the affinity for glucose is high. Okay, so given a choice, okay, when the action for if both these enzymes are present, let's say glucokinase and hexokinase both are present, okay, to act on glucose to glucose 6 phosphate, then it is the hexokinase which is going to take the preference. Okay, why? Because the affinity for glucose is high, it has a low Km value. Okay. So if you look at specificity, it acts on glucose, fructose, and mannose. Like it acts only on glucose. Okay. And what is more important is, even when the sugar level is low, glucose is utilized by the cells. Okay, so this is the importance. Now see over here, it acts only when the blood glucose level crosses more than 100 milligram per deciliter. Then glucose is taken up by liver cells by glycogen synthesis. Okay. So when the body glucose levels are low, okay, then hexokinase will act, okay, and glucose is utilized. Glucokinase becomes active when the blood glucose concentration is increased. Okay, so again we will discuss this when we talk about metabolism of glucose. So just now all you need to remember is that Km value denotes the affinity of the enzyme. Okay, so if the Km value is low, then it is preferentially it is more has more affinity for the substrate, and if the Km value is high, it has less affinity for the substrate. Okay. So we have covered all the important points. Okay, so thank you very much. Okay. See you with the next topic on enzymes.